Hello, everybody. I'm Edwin Ding. I'm with my partners Alex Lynn and Kevin Thomas. We just ask you guys to bear with us here. It's going to be a little longer than normal. But today we're presenting on the synthesis of platinum anti-cancer compounds. So first off, we want to give a special thanks to Dr. Rahman, Dr. Mukherjee, Dr. Shamik Das, Dr. Zhang, and Dr. Richards for providing us with a platform on which to work on this project. The first thing we need to understand is what actually is cancer. Now, I'm not going to sit here and insult your intelligence by telling you that cancer is a deadly disease, because we all know that. But it's a very specific type of disease, and understanding its cause helps us target better ways to treat it. So, as you all probably know, all of our cells contain important regulatory genetic material. This is basically information for our cells. It tells us what to do and when to do it. And the cells following this information then go on to maintain a delicate biological balance in what we think to be a healthy, functioning biological system. But once this intricate balance is disrupted, a lot of problems can start arising. So in cancer's case, the mechanism that is out of balance is the process of cell division. In our body, we have a specific need for a specific amount of any specific type of cell. And once that cell is operating on replication instructions that are corrupt and incorrect, you end up having cell division that's not within the normal parameters, and cancer is too much cell division. So here we can discuss malignant cells. So malignant cells are cells that A, do not stop dividing when they're supposed to, so there's too many of them in our body, and B, importantly, can metastasize, which means they can go to other areas in the body and cause harm there. And this is where we get the terminology for malignant tumors versus benign tumors. Now, benign tumors can still be harmful. It's a mass still. It can press on your bones and blood vessels where you don't want it to, but malignant tumors are much more harmful. And the development of these tumors can be caused by a multitude of factors. You can ingest carcinogenic compounds from cigarette smoke. UV light is another big one that causes skin cancer. And I'll just use skin cancer for an example. It's not like the rays from the sun are a vessel carrying this disease called cancer, and it beams it into our body. That's not how it works. Rather, it's the radiative properties of that light that go in and disrupt the connections within our DNA, leading to that corrupt genetic information and an excess of replication when we don't want there to be. So we found that cancer requires at least half a dozen mutations within our genome to actually become a thing. But when we consider how much genetic information we actually carry around, this doesn't seem like a lot. We have, every human has countless number of genes on their chromosomes, countless number of base pairs, and we find that a half dozen, unfortunately, it's really possible. And because the balance is so delicate, we find that things can go wrong on either sides of the spectrum here. You can have too many things, too many proteins in a cell telling it to divide, and an excess of those proteins would obviously lead to an excess of the cell because it's telling it to divide too much. But on the other hand, you can have not enough things on the cell. Hey, stop, we have enough of you. Not enough division inhibiting proteins. And that too would lead to an excess. And what makes this problem worse is that once you have a cell that's replicating off of the incorrect genetic information, the incorrect set of genetic information, if you think about it, it keeps replicating based off of this incorrect data, and its replicates are going to divide and with the same set of corrupt information, or even more corrupt information. So the problem really exponentiates. And my partner Kevin is going to talk about some of the cell lines that we encountered in our research, and some of the processes that we used in our lab to sort of deal with these cancer cells.
Yeah, so thank you, Edwin. So as Edwin mentioned, our lab primarily focused on cancer cells, and specifically we focused on pancreatic cancer and ovarian cancer cells. So some of the lines that we used include ASPC1, DANG, OV90, PANC1, and ovarian cancer cell line 82780. So as you can see in the top picture, on the left-hand side, we have the microscopic view of a normal pancreas, and on the right, a pancreatic tumor, and on the bottom, there is a picture of affected ovaries. And so, as you can see in both of the affected pictures, there's just a multitude of cells, and this is due to the cause of this continual replication and dividing of cells. But for us in our lab, the common feature between the cells that we used were they were all carcinomas, and so um, these cancers all occurred in tissues that line different organs. And so the unique thing about the lines that we use actually were that they weren't treated with any previous chemicals. So whatever culturing we did and testing we did, we could definitively see the results and effectiveness of our treatments due to the fact that these cells were not treated before. And so what exactly is a carcinoma? So like I said, a carcinoma is a cancer that begins in the skin or in tissues that line or cover organs. And so in our lab, we specifically covered two types of carcinomas. The first being epithelial carcinoma and the second being adenocarcinoma. And so epithelial carcinoma, this cancer occurs more in the soft tissue. And so this can include things such as muscle, fat, blood vessels, etc. Whereas an adenocarcinoma begins in a glandular or secreting cell. And so these include organs such as the lungs and the pancreas. But the good news is that both are treatable. And we can see that there's usually about a 50 to 70% success rate when treated. And this number obviously will increase with early detection and early treatment, just because of the fact that we're able to target the cancerous cells more efficiently and before it has a chance to really metastasize. And so, as you can see in the picture on the bottom, this is an image of a pancreatic cancer. It's affecting the pancreas, and where you see the two yellow arrows, that shows where the pancreas is affected. And so that's why these affected regions look inflated or a little bit bigger. And this is due to the fact that there is a growing tumor and an abundance of cells in those areas. And so a couple of different mechanisms we use to culture our cells is what I'm going to be discussing next. And so the first thing that we used was trypsinization. And the main purpose of this method is to disassociate cells using trypsin. So trypsin is an enzyme that helps our bodies digest and break down proteins. And specifically in the lab, we use them to prevent cells from sticking to the bottom of petri dishes and loosen them up for the purpose of transferring these different cells. And so a really basic process that we do for trypsinization is we suck the existing media out of the petri dish, and then we wash it with DPBS, which is a clear liquid, and it's a little salty solution that we use to just wash the cells. And we swish it around to make sure that all the cells are washed. And then we suck this DPBS out and add the trypsin. And once we do this, we'll put it into the incubator and allow it to sit for a couple of minutes. And after this incubation period is over, we'll take them out of the incubator and tap the bottom of the dish. And so that's what you see in the top picture. We get to see under the microscope these different cells floating around. And I think it's really cool because we're able to see these cells, these clumps of cells, just moving around in the trypsin. And that lets us know, hey, the trypsin is working, so you can move on. And so then we just mix the solution thoroughly with the micropipette, and we take some of this media with the trypsin, and we transfer it to new petri dishes with new media. And so this is just to make sure that we're not contaminating our cells and that the petri dishes stay sterile throughout our whole experiment. So next, I'm going to talk about a couple of cell viability tests that we were able to learn and use during our time researching. And so really a cell viability test is just a technique that we use to quantify the effects of various compounds on cells. And the basis of these kind of tests is to mix a known amount of cells with varying concentration of drugs, just to see how many of these cells are killed and we can use or are viable in order for us to use them in future experiments. So two of the effects that we really look for are the effects on proliferation and the effects on cell metabolism. So proliferation is just an increase in the number of cells, and this can be due to cell growth or cell division. And you can definitely see these in cancer cells because these cells are continually dividing and growing. And metabolism, as many of you guys already know, is just the set of chemical reactions that occur in a living organism to maintain life. And so a related thing is that these cells can be metabolically active while not proliferating. And so one specific type of this test is the MTT assay. And the MTT assay measures cell metabolism. And so this process is just the light yellow MTT that you guys can see in this picture getting metabolized over a week to about a week and a half into this dark purple colored solution, which is a formazine. And this process usually occurs in the mitochondria in cells. And so basically what this process looks like is we suspend the cells in a spheroid formation ECM. And this is just a mixture of extracellular matrix that promotes cell-to-cell adhesion. And so this is what we can see in a couple of days when we see these cells clumping together in spheroid formations. And so when we see this formation, we're able to know that it's time to add the medlum. And this medlum just contains cell proliferation modulating compounds, which increases the number of cells in the spheroid and allows them to get bigger and bigger. And so we let this process go for about 3 to 6 days, we can visually see that the spheroids are proliferating and expanding. And then after a couple more days, these cells reduce the initial MTT into this dark purple colored solution called the formazine. And we use this to quantify the cell number. And the instrument that we use to determine how strongly colored the solution is the spectrophotometer. And then another test that we use is trypin blue exclusion. And so this test allows us to measure cell proliferation. And it's able to work due to what happens when a cell dies. And so when a cell dies, the cellular membrane loses its specificity. And so this allows for anything to really penetrate the membrane and enter the cell. And that's what this trypin blue does. When the cell dies, the membrane no longer has a specificity, and the trypin blue can enter the cell and make them visible. And so from this, scientists can really count the number of dead cells or non-viable cells in the sample. And we're able to see how many of the cells we can actually use in the future for different experiments. And we can count these number of viable cells using this trypin blue exclusion. And so these are a couple of the techniques we were able to learn in the lab through our mentorship and through different research. But now my partner Edwin will go more into our specific mentorship, and he'll discuss PARP1 cleavage and the mechanism of cisplatin compounds.
All right, thank you, Kevin. So, yeah, first I want to discuss the PARP1 enzyme really quick. Unfortunately, due to external circumstances, we couldn't really get all the testing that we wanted in. But if we were to test, this would have been our method. So, PARP1 is an enzyme from a family of enzymes of 17 members. It's really important it plays the role of DNA repair in the cell. It repairs single-strand breaks by means of base excision repair, but basically you just need to know that it's really involved in the maintenance of our genetic information. And the cleavage of this enzyme is not really good news for any cell. So you can see that the cleavage facilitates cellular disassembly. Once PARP1 is cleaved, things start falling apart. But the convenient thing is we can use PARP1 cleavage as an indicator of the level of cell death in a sample, or the level of apoptosis in a sample. And how we do this is just to run a Western blot. So we would select the cleaved PARP1 out of our sample using an electrophoresis, cut out the section of the gel that contains the molecular weight of cleaved PARP1, which is 25 kilodaltons, transfer it onto a film, process it, and we would get these sort of bands. And based on the thickness of those bands, we can tell, okay, how much cleaved PARP1 is in our sample. And in turn, what does this tell us about how many of the cells have undergone apoptosis, and finally, the effectiveness of the compound that we're treating it with. So I'm going to move to talk about some of the platinum-based cancer drugs that we encountered. So the first to be discovered was cisplatin up here. And you can see that it's kind of a simple structure. It's platinum coordinated with 2 chlorines and 2 ammonia ligands. But after this basic structure was discovered, new ones soon followed. And it is effective. It has been shown to treat some cancers with over 80% cure rate. And a fun fact, it was inspired by mustard gas during World War I. So what cisplatin does is it enters this, the chlorine ligands leave, and the platinum binds to the actual DNA. So once it's bounded to the DNA, it inhibits the replication of that DNA and thus stops the cell from growing. And the same mechanism is assumed to be in place for the other compounds. And this is just more specifics about the mechanism. As you can see, it works on 2 fronts. It attacks nuclear DNA and mitochondrial DNA. Now, in the nuclear DNA pathway, what it does is it triggers a DNA damage response within the cell, which is usually responsible for repairing mutations, seeing what's wrong, seeing how it can fix it. But in this case, once it sees that the platinum essentially is stuck on the DNA and there's no way to remove it, what the cell decides is, okay, it's probably best biologically to undergo apoptosis instead of sitting here and wasting time and trying to fix this. So it triggers cell death. On the mitochondrial side, what happens is the binding of cisplatin, it triggers production of reactive oxygen species. And these are very damaging to the cell. These are ROSs. And that too can lead to cell death. But cisplatin is not all good. Just like all medicine, it has drawbacks, and the drawbacks of cisplatin are pretty serious. So in its administering, doctors have found that patients usually throw up 1 to 4 hours after they take it. So it's highly immunogenic. And because of the production of reactive oxygen species, it creates a lot of oxidative stress for our body, which means there's more harmful oxygen elements floating around that we can deal with. And at the same time, it deactivates our body's ability to deal with these harmful oxygen radicals, our antioxidant system. And this is what they mean when they say that a certain food is an antioxidant. Additionally, it's really taxing on our body's filtration system. It's really inflammatory to the kidneys in many cases. And the main thing is there's no factor on which it discriminates between the cells that we want to keep, the cancer cells, and the cells that we want to keep and the cancer cells. So it's an objective chemical pathway. There's nothing really telling it to say, okay, stop replication for these cells, but make sure not to touch these cells because these cells are the ones we want in our body. And to discuss, but luckily there are alternatives based on the viability of platinum as a sort of basis for these anti-cancer mechanisms. So my teammate Alex is about to go more in depth about this.
Right. So as Edwin and Kevin previously said, cisplatin it's quite effective against some cancers, but it's not really a cure for every type of cancer. The basis of our project was really the synthesis of novel platinum complexes and their characterization and testing them in cancer cells. And unfortunately, due to external circumstances, we weren't able to get much of the testing done, but we do have quite a bit of progress on the synthetic side. So to talk about the synthesis of these compounds, we need to understand some basic coordination chemistry. So all of the previous molecules that you saw, they're called coordination compounds or coordination complexes, and they're composed of two parts. There's a central metal atom, and then around the metal, there are molecules that are bound to it called ligands. And the metals will bind to ligands through the d orbitals. So there's 5 d orbitals. You can see a diagram of them here. And what actually happens is that if you imagine an isolated metal atom with no ligands around it, all of the d orbitals have the same energy. But when ligands approach the metal, the ligand orbitals will interact with the metal d orbitals, and because the shapes of the various d orbitals are different, the ligand orbitals will interact differently with different d orbitals and effectively split their energies. And typically, when we look at examples of coordination complexes, if you add up the number of electrons in the d orbitals with the number of electrons that the ligands have, you'll come out to about 18 electrons. So this is analogous to the octet rule for main group compounds, where you try and make every atom have 8 electrons around it. Well, for coordination complexes, you try to make the complex have 18 electrons. But there are some things about platinum which makes it special. For one, the example that we saw previously about the d orbital splittings, that is for an octahedral complex. But for platinum-2, which is what the majority of the platinum anti-cancer drugs are based on, they form square planar complexes. So the dz squared orbital just is lowered in energy. So they're from the octahedral example, but the general splitting pattern still applies. Now, for platinum-2 complexes, they're square planar, and they don't follow the 18 electron rule. They actually typically have 16 electrons. So if they have 16 electrons, they want to get to 18 electrons, they're electron deficient, which means that a characteristic reaction of these complexes is that they can undergo oxidative addition. And oxidative addition essentially means you add something to the complex, and in doing so, you oxidize the platinum, but also in doing so, you turn the complex into an 18 electron species. So you can see here cisplatin can undergo a oxidative addition to this compound, which is called oxoplatin. And this reaction can be done with other ligands other than the hydroxy groups. And then there's another thing in addition to electron deficiency that is characteristic of platinum-2 complexes, and that is the trans effect. And this is a kinetic effect that you need to take into account when synthesizing these compounds, because cisplatin, as you saw, It is square planar, but specifically the chlorines have to be on the same side and the nitrogens or the ammonia ligands have to be on the same side. If they're across from each other, that's another isomer called transplatin, and it doesn't work. It doesn't bind to DNA at all. It has absolutely no effect on cells other than maybe heavy metal poisoning. So you need to take into account the trans effect when synthesizing these compounds. And the basis of this effect is that some ligands will bind to the central metal stronger than others. So when you have a ligand on one side that's binding stronger to the metal, a stronger bond, like the definition is essentially there's increased electron density between the ligand and the metal. But if you have increased electron density on one side, then that means you're going to be drawing away electron density from the other side, thereby making the opposite bond weaker. So the ligands that have high trans effects bond strongly to the metal, so that when you attempt to substitute one of the ligands, the substitution will most likely occur trans to the ligand with a large trans effect because the opposite bond is very weak and it's easily broken, so you can substitute things. And this is a way that we can preserve or we can yield a single isomer instead of a mixture. So the first thing that we did is we synthesized cisplatin in the lab by ourselves, actually. And we start with a precursor molecule called potassium tetrachloroplatinate. And the reason why you can't just add ammonia to this and expect cisplatin to come out is due to the trans effect. So chlorine and ammonia have very similar trans effect levels, which means if you just straight up added ammonia to potassium tetrachloroplatinate, you would get a mixture of the cis and trans isomers, which wouldn't be very good because then you would need to separate the isomers, which isn't very easy to do, but also because that would kind of be a waste of starting materials if half of your starting material ended up as a product that was useless. So to combat this, we substituted all chlorines for iodide ligands. And because the iodide ligands have a much stronger trans effect than chlorine ligands, then when you add ammonia to the tetraiodide, it substitutes trans to iodine, which will always give your ammonia ligands in the cis position. Then using silver nitrate, we can precipitate out silver iodide, and then finally using some form of chloride, we can make cisplatin. So the main thing for our research, there were 3 synthetic targets that we tried to hit. So the first, we wanted to make tetrachloroplatinate, which is the precursor molecule you see here. We wanted to make it undergo oxidative addition to make a platinum 4 complex, where it has 6 ligands around. And the top and bottom one, the OHs, those are called axial ligands. And we tried to make them either hydroxyl groups or acetate groups, which are water soluble. So this would increase the water solubility of the compounds that we produce. The other one is that we tried to make dialkylamine oxalato complexes. So essentially make compounds analogous to cisplatin. And then the last one is instead of using free ammonia or amine type ligands, we would use substituted by pyridines as the nitrogen containing part. So for the first synthetic target, the general method that we utilized is to either to dissolve the precursor molecule in a solvent or create a suspension of the precursor molecule in a solvent. Typically, this was done with water, but we could also do it with ethanol. And in fact, we could also do it with acetone for some of the more water insoluble precursor compounds. And to that, we add about a 50 to 150 times molar excess of hydrogen peroxide. And what this does, hydrogen peroxide is an oxidizing agent. So it'll oxidize the platinum from platinum 2 to platinum 4. And in doing so, it will undergo that oxidative addition reaction to form the complexes that we want. So you can see the percent yields here are pretty decent. In comparison, when we made cisplatin, the percent yield was about 83%. So these are pretty good percent yields for this reaction. Another thing that is you have to take into account with these reactions is that when hydrogen peroxide oxidizes the platinum, it itself gets reduced to water and oxygen. So the reaction foams quite a lot, and we do these reactions at increased temperature, about 50 to 75 degrees Celsius. So once in a while, you have to take the cap off the reaction vessel and let some oxygen off. Otherwise, the reaction bubbles and there's increased pressure inside the reaction vessel. There's a possibility of it exploding. And so the second synthesis target that we wanted, this is more of a multi-step synthesis, and we haven't actually completed the synthesis in full, but we have tested several steps in it. Most notably, the first step, we react tetrachloroplatinate with excess dimethyl sulfoxide in water, and we get about a 94% yield of the cis dimethyl sulfoxide dichloroplatinum. And from there, we can use silver oxalate to precipitate out the chlorines and replace those chlorines with an oxalate ligand. And the reason we do this is because even though cisplatin has those chlorine ligands, some of the new drugs, they are using oxalate ligands instead of chlorine because they found that the oxalate ligand is actually more stable within the body, and it doesn't have as many side reactions with other proteins and molecules that are found within cells that sometimes deactivate cisplatin. And then from there, the dimethyl sulfoxide ligands are directly substituted with pretty much any amine of our choice in water. The first substitution occurs around 40 degrees C, the second one around 100 degrees C. So it's possible to put two different amine ligands on there. So we can treat it with one equivalent of, say, isobutylamine at 40 degrees C and complete that. And then we can isolate that and then treat it with another equivalent of, say, of methylamine at 100 degrees C, and that would complete the second substitution. So we have a lot of variability in which amine we can substitute onto. And finally, the last synthetic pathway that we used was using substituted by pyridine. And this is the easiest out of all the syntheses because essentially we just add a bipyridine to a solution of tetrachloroplatinate in water, and we just let it react under stirring and about 75 degrees C. And after about 10 minutes, the reaction's done. All you need to do is filter it off. So you can see At the bottom there, we have all of the substituted bipyridines that we were looking at using. Due to time constraints, the only one we were actually able to do the reaction for was the bottom left, which is the dimethyl substituted bipyridine. This forms kind of a really bright yellow complex, but it hasn't been isolated yet, still in solution. And so for our future work, we plan on, or at least this project will have further synthesis of novel platinum complexes. And we are thinking about directing the syntheses towards the synthesis of some novel heterocyclic ligands. So the substituted bipyridine ligands that we saw were purchased from Sigma Aldrich, but we're looking to use some precursor molecules to make our own for this purpose. We also seek to characterize more novel platinum complexes through mass spec, NMR, IR, and for those that we can get crystals for, as X-ray crystallography to confirm the structures, and also testing of those novel platinum complexes through the techniques mentioned earlier, such as the trip and blue exclusion and MTTSA and things like that.
All right, good job, guys. Does anybody have any questions?
Yeah, I have one. Okay, so hi, sorry, I'm David. My question is there? I don't know if you guys covered this. I might have missed it. Is there a particular reason that you guys used platinum-based cancer drugs? Like, were these drugs more effective to the cells that you used, the cancer cells that you used, or was there like another external reason? So yeah, that was my question.
Yeah, that's a good question.
Okay, Alex, do you want to take this one?
Yes, yeah, I'll take that one. Yeah, that's a good question. The reason is that platinum complexes, I mean, as Edwin mentioned, this kind of cell toxicity of platinum-2 complexes with cis ligands was actually inspired by mustard gas because it operates off of pretty similar mechanism. But we find that platinum group metals just have a characteristic of being square planar, whereas a lot of other metals, they form octahedral complexes, which just for the reasons of how the DNA, the nucleotides in the DNA is spaced, isn't good for binding to it. So the square planar structure allows platinum to bind to DNA most effectively, even more effectively than other platinum group metals such as palladium. So that's the reason why we chose platinum instead of other metals, just because it binds to DNA the most effectively.
Yeah,
Okay,
and
cool.
another thing is we can, people have used gold before in place of platinum, and that works in a different sort of mechanism, but we decided to go the route of taking a known anti-cancer compound, this platinum, and sort of mitigating or attempting to mitigate the harmful effects that we saw previously. So we have a basis on which to work off of, and we just keep editing the same template. Essentially, we keep making changes to that cisplatin template, and we thought that was the best way to approach improvement.
All right, cool. Thank you. That answers pretty well.